So here we go, the sheep got the glory, the pigs are now doing all the work. If you listen to the talks before, you might not think about that, but I will give you my logic for it. So, of course, it's a great pleasure to be here again. I have been here in Scotland for quite a long time. Now I live in Munich, and Munich is twin city with Edinburgh, which I think is very fitting because if you look in both cases, the males have their particular way of dressing. And of course, in Munich at the moment, the lederhosen are really very much in fashion because this month we will start also the Oktoberfest. And at the Oktoberfest, you have, of course, a huge consumption of beer, but not only beer, but also many, many Schweinshaxen. And so with the Schweinshaxen, so pig hocks, very nicely, I come now to the animal models. Of course, the pig is a very important agricultural model, especially in Germany. Maybe here the sheep is more important. And then, of course, and this is what I will be talking about, the pig is also an important model for biomedical research, and particularly for disease models, and also xenotransplantation. So I will mainly concentrate on that side of it. So why are we talking now about it? Why didn't we talk years ago about it? For a long time before Dolly, it was actually only possible that we do genetic manipulation, precise genetic manipulation, to make animal models in the mouse where we had the embryonic stem cells. And Ian mentioned that right at the beginning in his talk. Since Dolly, we can actually also put defined genetic modifications into large animals. And so which large animals would you then use for the animal models if you look at human diseases? Well, you want some animals which are anatomically and physiologically similar to humans. They should have a similar size so that you can also use human equipment for the surgery, for radiation treatment, for imaging technology. So it's not only that you have a model for the disease, but also for disease treatment and also for the development of technologies which have to do with the healthcare system. You can do, of course, because they have a longer lifespan than mice, it's larger, you can do longitudinal studies. That means we can take right at the beginning, after birth, we can take samples, we can take continued blood samples, but also tissue samples, which because of the size you can't do easily in the mouse. It has the disadvantage, of course, that our experiments take quite a long time. They have similar pharmacokinetics. You can do it in the mouse by introducing or deleting some of the genes. In the pig, it might not be necessary. There are differences in the immune system, but there are also similarities to humans. And if it comes to pigs, so a lot of it might also apply for maybe the sheep, but if you come to diseases which might also have to do with diet, the pig you can feed a human diet, cafeteria diet if you want it. They have a large litter size, so you get lots of animals born. And of course, a pig, unlike the sheep or so, you can keep it indoors, so we can keep them in an SPF facility if it's required. When these experiments started, the question was, well, is it actually an improvement? Will the pig models be any better, different, or just the same as the mouse models? And I will give you some examples for that. So here is the logic again, why not only the mice? If you already have the mice, why do we need a pig? So the mouse is still one of the most important animals to do the basic research in it, either in cell culture first of all, then you go into the animals models. But a lot of the experiments, and especially the drugs which have been tested in the pig, fails then on the way into the clinic. And so here's the success rate. At some point it was estimated it was only 1 in 5,000. Might also be something to do with the mouse models and people rethinking the mouse models, how it has been treated. So the idea is that you might need something in between to sort of bridge this gap from the bench to the bedside, that maybe a large animal models for preclinical testing, for validation, for safety of new technologies would be very important. And here, you don't have to read the table, and the table I should probably update is the large number of experiments which have already been done in the pigs to make animal models for different types of human diseases. There are several groups working on cancer models, cardiovascular diseases, the diabetes. This is probably one of the best characterized models. Together then we also have the neurodegenerative diseases and other models which are very important is, for example, cystic fibrosis, one of the most common inherited diseases. Again, a very well-defined animal model. We have Duchenne and muscular dystrophy and many others. And now with the new technology, many more will also come on the scale. our own lab, and that's what I will be talking about now, we're looking into cancer models, and I know there's also interest here at the Roslin Institute. And here again, you could also argue, why do you have to make new transgenic models or genetically modified models if there are already perfectly all right models for there? For example, the dog, which has a very increased cancer rate. You can see here, like the melanomas are highly increased, osteosarcomas. But of course, these are sporadic cancers, and working with dogs, unless it is a patient, is very difficult. And that's particular because it is a pet. So we have a different view if it is a pet or if it is a food animal, which might not be quite logical. So here the livestock animals, they get cancer very rarely, just like in humans really. Some inherited melanomas we have found. We have in early pigs, we can see some lymphomas and urinary system tumors. But other than that, especially the pig goes to slaughter with 9 months, so it doesn't really have time to develop much cancer. We can transfer into the pig cells which has been transformed, and we know that then the cancer cells can work, and of course we can make the genetic models. And here's the first example. This was done by simple gene targeting, and we have heard in the talk before from Chris that it is difficult, but I can tell you it can be done. So here you have the colorectal cancer, you have the polyps here which develop early, and this is actually a pig. So what is the reason why they develop? It is a gene which is called the adenomatosis polyposis gene, the APC gene, which in 85–95% of all cancers have been mutated. And you can see here you have the hotspots of mutations. On the top you have for sporadic cancer, on the bottom for those which has been for inherited cancer. And here's one, this large line is a mutation in humans which is 1309. So it's a very common one, and it's also quite severe. If you introduce this mutation into the pig gene, do the gene targeting, nuclear transfer, produce a pig, you have then animals which look like that. So they get a large number of polyps in their gut. If you leave the animals a little bit longer, you can see here the tumors increase in size. This is a carcinoma in silico. And what has been nice is that you can see that all the polyps develop in the large intestine, and this is exactly what you also find in humans. If you would do the same mutation in the mouse, then you would have it in the mouse in the small intestine. So this was the first indication that the same mutation in the pig gives us the same phenotype, and the mouse is slightly different to this. We can then, of course, analyze on molecular basis if our models replicates the disease. Here, for example, is a human cell line which has a very similar mutation. This is one is 133008, and you can see it is a truncated APC protein which is now being produced. And here are our we have two different pig models. One is a 1309 in pig 1311, and another one is 1061. Both of them have the truncated protein. Before you will have cancer developing, you have a loss of the second allele, and you can see this here if you look at the polyps. Here we have no more wild-type allele, we only have now our mutant allele. This has then the effect that the Wnt pathway has been disrupted. You have an accumulation of beta-catenin, and you can see this here very nicely in the crypt. Here's beta-catenin all over the cells in the cytoplasm and the nucleus compared to the wild type, and these cells proliferate further. You can see this here by the Ki67 staining. And of course, you could also see it that we had all the polyps. So what can you do once you have an animal models like this? We can, of course, do transcriptome and genome analysis to see what genes, how they change over time during disease progression, and also susceptibility. We have collaborations where people are now looking at the microbiome in the pig and to see also how the microbiome alters or maybe also influences the disease status, and maybe that in combination with a diet. We're looking at biomarkers. We look at the influence of inflammation, because it is clear that the immune system plays a very important role in the progression, but also in the regression of some of the polyps. We have done the first intervention study, small drug with sulindac, another one with a diet is following. And we later will hear about talk about organoid cultures. it is quite important that if you want to do drug studies, that you can do first tests in tissue culture and then afterwards go into the animals. So we are now also developing organoid cultures from the porcine model. I showed you that it took about 20 months until we had our first sort of carcinoma in silico. To make it faster, we're now introducing more mutation, for example, this KRAS mutation, which is common in adenocarcinomas, and often you also see p53 mutation. We can introduce others. And to just give you a couple of examples, here you have some biomarkers. What you can see here, this is microRNA, where we have taken the blood samples and looked in the blood serum sample. If you can see a difference in the microRNAs, you can see here, these are our wild-type controls. These are animals which have low polyp numbers. And then here you have animals which have high polyp numbers. The same here for the miR-143. High polyp number, we can see a slight difference in the microRNAs. We still have to confirm it, of course, with more animals. We can look at the disease progression, and that's something which is difficult to do in the mouse because you can't take repeated samples in the mouse, and in the human it's for ethical reasons difficult. But here, what you can see here, you have a difference in the heat map between the low-grade interepithelial neoplasia and high-grade interepithelial neoplasia. And this is done now by laser microdissected samples. If you don't laser microdissect, we get a slightly different pattern because we see also a lot of immune cells. And this one here is something which we're particularly also interested in, but we have to do more work on it. In our animals, because they're outbred, we have a difference in the disease phenotype. We have animals which have, and these are siblings, which have low polyp number and high polyp numbers. And you see the same also in human families, in human patients. And so the question is, what is the other driver gene? So there must be other genes involved. And so again, we have done a transcriptome studies, but this time not from polyps, but from healthy tissues around it. And then just see, do we have genes which cluster? And if they cluster, can we see, are they clustering with high and low polyps? So there's still more work to be done, but this part here are mainly the low polyp animals. So there is definitely a clustering, and now we have to look into those genes, what that could mean. So here's another example for the imaging. If you have an adenoma or an adenocarcinoma, they quite often overexpress the CCK2 receptor. And of course, you can use then CCK2, labeled CCK2. And in this experiment, unfortunately, you can't see it here in the light very well, but you see sort of a little bit of blue. And if you would look here, you would see there is a polyp. And simply during the endoscopy, this was sprayed during the endoscopy onto the gut and then seen where would the labeled CCK2 bind to the receptors, and you could really see that it was binding on top of the polyp. So, and with this, I switch over to one of the other animal models. As I mentioned, we're also interested in KRAS. We're also doing p53, and p53 is one of the molecules which is mutated in almost 60% of the cancers. And you have a human disease, which is called Li-Fraumeni disease, where you have p53 mutations. And in the p53, you can either have a loss of p53 function, or you can have mutations, and some of the mutations have dominant negative functions. One of those is this mutation here, which is a substitution at position 157. And this is then affects the DNA binding. It inhibits the wild-type p53, which might still be there, and it might advance angiogenesis, metastasis, and tumor invasion. This model also was still done by the old-fashioned gene targeting. And what we had to do here is we had to introduce two different inserts. In one, we added a lock-stop-locks cassette into the first intron and then the point mutation into the fifth exon. So this works. This lock-stop-locks cassette can be removed by Cre recombinase, and then you express the dominant negative mutation. So this is quite commonly done in the mouse. We had shown here for the first time that you can only do it in the pig. We can have shown in vitro that this mutation is functional. It overexpresses then the mutant protein, and you also have then cells which are almost chemo-resistant. What I want to talk about to you now, or give you the example, is if you leave these cassettes in, you principally also have a knockout. So we can either look at knockout animals or at the mutant animals. And we were wondering what these knockout animals, either heterozygous or homozygous, might have as a phenotype. In humans, mutations, they're usually heterozygous. You get soft tissue sarcomas, you get osteosarcomas. In females, you will also get breast cancer. In the mouse, for heterozygous knockout, you get often osteosarcomas with about 10 to 18 months, and the full knockout gives you then mainly lymphomas. So what does it do in the pig? We see in the heterozygous animals at roughly at 2 years. So the first years we don't see anything, but between 20 to 24 months we can see that they all get osteosarcomas. In our case, at least all the animals we looked at so far. So we're not too far off from the mouse experiments. And when you then come to homozygous animals, we can actually see tumors between 4 and 8 months. And again, we have osteosarcomas. And this timing is very similar to what you actually see in the mouse, which was a little bit surprising. In humans, you have two time periods where you get osteosarcomas. It is in the young people when the bones are growing very well, or in the older people. And so here we probably have a model for the juvenile osteosarcomas. So here's one of our pigs when it goes into the MRI and PET scanning for the tumors. And when you look then afterwards at the bone, this is an animal which was 7 months old, which we had to sacrifice because it wasn't doing too well. And you can see here, this animal had multiple osteosarcomas. You can see a tumor here, here, here. It's growing together, and you have another tumor here. And as you would expect for P53, you can actually see that you have very large chromosomal abnormalities. You have giant nuclei, multinuclei, micronuclei, so all the things you would expect. So I mentioned just briefly before that we can also activate mutations. We also have animals which have a KRAS mutation. To activate it, we just have to add CRE. So now we can combine KRAS with P53 or KRAS with APC, and those animals have also been born. And principally where the tumor develops depends now on the CRE. To see where we can have CRE expression, we have also made reporter pigs. These reporter pigs have a double reporter which expresses the tomato gene, and this tomato gene is FLOX. It has been inserted by homologous recombination into the ROSA26 locus, and if you then add CRE, it switches to green. And you can see here is an animal which is expressing villant CRE, and here's the gut, and you can see that in the gut, in the crypt, we now have the green fluorescence, while the rest is still red fluorescent. So it seems to work in the animal, but other researchers have also shown that you actually don't have to do it in the animal. You can just take out some of the tissue samples, activate your mutation, and then put this tissue sample, principally the cells, back, and then the tumors will also develop. So that saves us making another whole animal. What you can, of course, also do now with the new technologies, you can probably do local application of the CRISPR-Cas9, and that's something we are interested in. So we want to go now back into the gut where we already have the polyps, you have seen it, and then either with electroporation, because you can grip the polyps, or with injections, we would like to add now CRISPR-Cas9, and then you can actually use it to activate our CRE, but you can also use it then to knock out P53 and other tumor suppressors, MAT4, and so on. So with this, I will stop for the tumor models or for the disease model, give you briefly one other example, and this is also the pig working for xenotransplantation. So here you have, of course, the pig with all the choice cuts. But as I said, we can also use the organs to actually save lives. So not just for nutrition, but maybe to save life, and there is an organ shortage. So the organ is rejected. If you take a pig heart into humans, it's rejected within minutes. So what you have hyperacute rejection, you have acute vascular rejections. We have to do multiple genetic manipulations. So here you have a whole list of transgenes, several complement regulators, and we have to inactivate several of the genes. I'm a little bit sorry, some of the text has sort of moved around, moving it from the Mac to the PC. So we got involved in it about 4 years ago when I came to Germany, because there is a large group who worked on xenotransplantation. One of the people is also here. This is Heiner Niemann. So the question at the time was, do we get animals which have high CD55 expressions, which is one of the very important complement regulators? So that was one of our tasks, get multiple genes in, all the multiple genes at one locus, so that when you breed the animals, they don't segregate, because otherwise you have a huge breeding program. High expression CD55 at least, and possibly then also the knockout of the GAL and the CMA epitopes, which are all sugar epitopes, which, like was mentioned before, they might be why some of the recombinant proteins from animal cells might not be working so well. So we started this in two different ways. We thought, okay, we know the ROSA26 locus should express transgenes, so can we place all our transgenes into the ROSA26 locus? So we targeted first the Hemox gene into the ROSA26 locus. We made an animal and could see that we had Hemox expression in every single organ. We then took cells from this animal and retargeted it with a mini gene from, which expressed CD55. And surprisingly, because other people had already made animals with a mini gene, with multiple copies as a transgene, the single gene at the ROSA26 locus gave us really, really high expression of CD55. And that is, I mean, I don't have a comparison to the other one, but it is quite high, even though it's also high. So we have high expression of CD55 with a single gene at the ROSA locus. In parallel, we had also done experiments where we thought, okay, mini genes hadn't been shown to be very efficient so far, so maybe we also work with genomic genes because the genes are big. We used BAC vectors. So we used the BAC vectors for the CD55, for the CD59, and for the CD46. We then made them a little bit shorter by recombinating, added a couple of other transgenes to it, like the Hemox and the A20, then introduced all these genes together into cell lines, tested those cell lines for some cells which really express CD55 very high, and if possible, all the other transgenes. So that was a minimum of almost 400 kilobases. And we now know in these pics which you see here, we have almost a megabase on transgenes integrated, all the transgene at a single locus, and they really also have very high CD55 expression. A little bit less than here, but higher than anything which had been expressed. So is it all functional? Here is a test of the cells where you challenge them with human complement. The wild type cells here will lyse. If you then have cells which only express CD46 and CD55, we had some effect, but with all genes together, we couldn't get any lysis. And actually, we get the same result also for these cells which really highly express the CD55. So then the next one was to the inactivation of the GAL and the non-GAL cells. So we had now the multi-transgenic cells, and we also worked with some wild-type cells. We also sent some of the wild-type, the multi-transgenic cells to Heiner Niemann's group. He already had a zinc finger against the GAL epitope. He inactivated the GAL epitope. We got animals. And in a separate experiment in our lab, we used CRISPR-Cas9 and just try to do two inactivations at the same time, the CMA and the GAL epitope. And we had animals born, and you can see here in those animals, if you look at the organs, there is no more NOI5 GC. So here you have now a combination between cloning, genome editing, and also production of transgenic animals. And my final words, really, the conclusion is, for the large animal models, there is definitely a clinical need. And more people now with the technology are improving, will be making more and more of those genetically modified animals, either by genome editing or nuclear transfer combinations. The pig is the choice for the xenotransplantation. And the improvement which have been done over the years now allows that organs can actually, in baboon experiments, survive for almost 2 years. So I hope I could convince you, yes, the pig is a very hard-working animal, especially in the area of biomedical research. I just wanted to mention this also. Lots of people are working on genetically modified large animals. We would like to avoid that we all do the same and duplicate too much of the work. And so there is an EU cost action, which is led by Eckhard Wolf from Munich, where we try to at least coordinate our efforts and also the way we analyze the animals. Here are the collaborators. These are people in my groups. These people are already left. Professor Fries has done the transcriptome analysis. We have collaboration with the Helmholtz Zentrum on the genome editing, and of course with a lot of the medical people from the Klinikum Rechts der Isar in Munich. Eckhard Wolf's group helps us with the nuclear transfer, and so does the group from Heiner Niemann. And yeah, this is different projects I didn't talk about, and this is our funding. And thank you for listening.
Do we have questions for Angelika? We've got one up here.
When you thinking about xenotransplanted tissues or organs in humans, and maybe there's evidence already from the macaques, do you expect that there will be host cells will begin to infiltrate the transplanted organ and that it will gradually return to being human, or do you expect it to sit there being a pig tissue in a human being for the rest of the life of the recipient?
It probably depends what tissue you will be working with. In some, like kidneys, there are experiments where at least the blood vessels might become human, or if you implant it earlier, I wouldn't expect that the whole tissue will change. In other experiments, probably the ones which are closest to the clinic are the islet cells. These will be encapsulated, so no. So no, principally, I would think it will stay a pig. What you will have infiltration will be probably also immune cells, which you might not want.
Angelika, thank you. Wow. Everything but the kitchen sink. But you missed one thing, and that's the possibility, touching upon the last question, about introducing human cells into the organs that you might be creating, to say, create a more human vasculature, for example, that might come by an early introduction of lineage-restricted stem cells. Any talk or action in that capacity in the field?
The group is working on xenotransplantation. There is one partner who is working on that and tries if you can entice the human blood cells into the blood vessels of the organs. And there have been experiments, as I mentioned just now, that if you implant very premature kidneys, just the kidney Anlage, then the blood vessels actually come in from the host. And so you would then, and so that the kidney develops in the host for a while, and then you would have already the human blood vessels. But yes, people are talking and trying it out.
Is there another question? We've got one, Alison.
Hi, thank you. So obviously the main part that kind of stopped the research into xenotransplantation some time ago was the porcine endogenous retrovirus. But now having the CRISPR-Cas9 technology, I think it's possible to kind of remove that problem. Has it been resolved, or is it still a big issue that is a barrier in that field?
I mean, first of all, there isn't a lot of evidence that the porcine retroviruses actually infect human cells. So it's not such a dramatic risk, first of all. What I haven't mentioned is the cell line we're working with is actually free of perf-Cs. So we do not have any perf-Cs in those cells. And then, of course, there's an experiment now from Church's group who has shown that theoretically you can also knock out all the perfs. And of course, there are repeated experiments of that. We have to see if you get live animals.
Okay, thank you.
Any other questions for Angelika?
I enjoyed your APC model in pigs, and our interest is in colon cancer, but have you, is there any evidence of any genetic variation in expressivity or whatever of the main allele? For instance, mouse is obviously the MOM1 allele. It was picked up by Bill Dovick et al. There's very little evidence of any genetic modifiers of the APC allele because it's such a powerful allele in humans. But have you seen any evidence from breeding stock of variance in the expressivity?
We are looking at allelic expression. We find difference in allelic expression in some of the animal, but not the APC gene.
One down the front here.
So using a pig for as a nice cancer model, as you've shown, will be very relevant for coding sequences. But there are control elements that are also involved in controlling the expression of these genes, which probably are not as conserved between the two species. How do you think you're going to approach that to model a cancer that's from a gene control element?
It is at the moment, but here I'm relying on the work at the Roslin Institute that we have more information soon. So when we look at the microRNA, it's difficult to interpret because lots of the microRNAs, I was told, are not annotated yet for the pig. So that is true for some of the non-coding RNAs, but with the sequencing, we of course get both the coding and the non-coding, and we also did the sequencing, so we have the microRNAs. So yes, we still need more information so that we can make conclusions. And some of them are quite conserved, like the ones I showed you, but
Okay, with that, I think we should thank Angelika again for telling us about these leading cancer models. Before I release you for lunch, I think we should all thank all 4 speakers today and also show our appreciation to the 2 gold sponsors, Recombinetics and Exemplar Genetics.